Good morning, students. Sairam, myself Sridevi Rao will be starting a new lesson today. Hope all of you all are fine. So we have finished with 3 lessons. Now let us start with the third lesson today. Life processes in living organism part 2. Okay. Now this lesson we are going to learn about life processes. You all know already what are life processes. All the processes which take place in our body like digestion, respiration, reproduction, circulation, all these are called as life processes. Okay. Now today we are going to learn about reproduction in this session. Okay. Now what is meant by reproduction? Formation of new organism of same species is called as reproduction. So you all know the formation of new organism from the existing one is called as the reproduction. It is also one of the various reasons responsible for evolution of each species. Only because of reproduction, all the organisms we all evolve only because of reproduction. Okay. Now there are two types of reproduction. We are going to learn about asexual reproduction as well as sexual reproduction. First we are going to learn asexual reproduction in plants. Okay. Then we are going to learn sexual reproduction in plants as well as sexual reproduction in animals. We are going to learn all these. We are going to learn in this lesson. First we will start with asexual reproduction. What is meant by asexual reproduction? Formation, the process of formation of new organism by an organism of same species without the involvement of gametes. That means there is no involvement of male and female gametes. The formation of new organism without the involvement of gametes is called as asexual reproduction. What is asexual reproduction? It is the process of formation of new organism by an organism of same species that is important, without the involvement of gametes. Okay. In sexual reproduction we'll tell there is involvement of gametes, male gamete and female gametes. But in asexual reproduction there is no involvement of gametes. Okay. As this reproduction does not involve union of two gametes, the new organism has exact genetic similarity with the reproducing organism. That means the parent and the offspring will look similar. There is no genetic differences. There is no changes in the parent and there are only the small one. That will be the parent and the daughter cells or the offsprings will be similar. Okay. Because there is no union of, we saw in meiosis how two chromosomes are crossing over takes place and there is genetic variation, but there is no involvement of chromosomes, or there is no involvement of two gametes. So only from the parent it is divided into smaller parts, that is called as the asexual reproduction. This is uniparental reproduction and occurs by mitotic. We learnt. Okay. So this is a there is no involvement of two parents, that is male and female. Only single parent reproduces, that is by mitotic division. We learnt in mitosis how first the nucleus divides, that is karyokinesis and cytokinesis. Similar, just there is division of the parent cell, that is this one. What is this? Asexual reproduction. Now, coming to the asexual reproduction in unicellular organisms, first we will see first we will see binary fission. Okay. So this, what is meant by binary fission? First, this when you take stagnant water. Okay. When you take stagnant water and keep it in your house, or take little bit of aquatic plants and or wheat and put it inside that stagnant water and keep it for 2 to 3 days till all the plants get decomposed. After that, take 1 drop of that water and keep it under the microscope, and when you observe, you can see the paramecium, that is the unicellular organism called paramecium. It will undergo binary fission. So what is meant by binary fission? In binary fission, the parent cells divides into two similar daughter cells. There is no involvement of gametes. Only the parent cell will divide into two different daughter, similar, both will be similar. And many, it is seen in many of the prokaryotes like bacteria and protists like amoeba, paramecium and euglena. Okay, so where it is seen in many prokaryotes like example is bacteria and other protists like amoeba, paramecium and euglena. In this lesson we will see the examples of amoeba, paramecium and euglena. Okay, so that is what is meant by binary fission. The parent cell divides into two similar daughter cells. Just a division of the parent cell is called as binary fission. Okay, even in the eukaryotic cell organelles like mitochondria and chloroplast also, they perform this asexual reproduction by binary fission. Not only prokaryotic cell, even in the eukaryotic cell organelles like mitochondria and chloroplast. Okay, they also perform the asexual reproduction by binary fission. Binary fission occurs by mitosis. Mitosis, we learned mitosis or amitosis. What is amitosis? It is just a division of the cell. There is no formation of spindle and no chromosomes are involved. Amitosis means just a division of the parent cell. Okay. Now, coming to how the axis, that is the division of different is different in different protists. That means how they divide. Sometimes you all know amoeba does not have any particular shape. So the division in amoeba, that is binary fission in amoeba, it can take place in any plane. Okay, the amoeba has got different any shape, so anywhere it will divide into two parts. So in any plane it can divide, and that is called as the simple binary fission. Because it does not have any particular shape, it can divide in any particular manner. You can see any way it will divide. See, this is the amoeba, and the projecting the shape that is called as the movement. It moves with the help of pseudopodia, that you all know. See how in the second picture how the nucleus becomes elongated, and the third picture you can see how it is constricted in the middle. Okay, it's going to now two daughter amoebas are formed, which is identical to the parent amoeba. This is simple binary fission, where the axis, it can be in any plane, it can divide anyhow. Okay, this is in amoeba. Now coming to paramecium. Paramecium is like a slipper shape. You all have seen paramecium, we have learnt in 8th standard also. What a paramecium, we have drawn the diagram of paramecium also in your notebooks. So paramecium, I'll show you the picture of paramecium. Paramecium divides by transverse binary fission, whereas euglena by longitudinal binary fission. So we'll see paramecium, how it divides by transverse. That means divides into in this manner, in the horizontal manner, and euglena in vertical manner, that is in the longitudinal manner. Binary fission is usually performed by living organisms under favorable conditions, that is when they have abundant food material, so they will divide. So amoeba will divide in any manner, anyhow they will divide. But when you come to paramecium, they will divide in the transverse binary fission, and in euglena it is longitudinal binary fission. So paramecium, it is transverse in the horizontal way, and euglena it is in the which way? Longitudinal way. You can see here. See how paramecium, it is transverse binary fission. Transverse binary fission means you can see that is the parent paramecium will slowly get in middle in the half, it will divide the transverse direction, and two daughter parameciums are formed. That is just by dividing the parent cell into two. Okay. In euglena, you can see here, euglena has got a flagella. You can see this euglena. You can see the next stage how it has become. Then the new inside the nucleus, it will divide into two, and then there will be two flagella. It will be together for some time. Then it will divide from its parent, and two daughter cells from the parent only it will divide into two. Okay, so we saw paramecium and we saw euglena. First we saw amoeba. In any plane it will, that is simple binary fission, in any plane it will divide itself. And paramecium we saw in the transverse, in the longitudinal way it will divide. And we saw in euglena in transverse in paramecium, and in euglena it is longitudinal binary fission. Okay, understood children how binary fission is taking place? Now let us come to the next part. It is multiple fission. So we saw you binary fission. One we saw in amoeba, we saw in paramecium and euglena. In amoeba, how it is dividing? That is simple binary fission. In any manner, it will divide into two in any manner. Then we came to Paramecium. I told you in paramecium it is transverse binary fission, that is in the longitudinal way, and in euglena it is, what is the method called? It is in the longitudinal way, like this method. Okay, so there are we saw about the three methods of binary fission in amoeba, paramecium and euglena. Now coming to the next part is your multiple fission. That is, this is also asexual reproduction in unicellular organisms. Next one is multiple fission. This is asexual reproduction by multiple fission is performed by amoeba and other similar protists. In amoeba, okay, in some unfavorable condition like lack of food or any adverse condition, this amoeba will stop formation of pseudopodia. What will happen in favorable conditions? We saw it will divide into two in any plane. But in unfavorable conditions like lack of food or any adverse condition, what will happen? The amoeba will stop the formation of pseudopodia and it will become round in shape. Okay, the pseudopodia is like finger shaped, that it will stop and it will form, it will become round in shape, then the movement will stop. Okay, so what will happen in this? It becomes rounded and forms a protective covering around the plasma membrane. There is a covering around the plasma membrane, and such encysted amoeba or any protist is called as a cyst. So what happens? In under unfavorable conditions like lack of food or any adverse condition, they will not form the pseudopodia, so they will be stagnant, they will be in one place and it will become round in shape, and they will form a protective layer around it, which is called as the cyst. And many nuclei are formed by repeated nuclear divisions inside the cyst. So inside the amoeba itself there will be multiple nuclear divisions. There will be continuously dividing, and it is followed by cytoplasmic division. So inside that cyst itself many amoebulae, that means small amoeba will be formed. That is, nuclear division also takes place and cytoplasmic division takes place inside the cyst. They do not come out of the cyst. It is a protecting covering around the amoeba. Okay, so they remain uncysted till there are adverse conditions. So there is lack of food or any other adverse condition. They will remain inside the cyst and they are living inside the cyst. When there is a favorable condition, the cyst break opens and the arrival of favorable conditions, and many amoebulae are released. So when the favorable conditions come, the cyst opens and you will get many amoebulae. The small ones of amoeba is called as amoebulae are released. So what happens under, you can see here. So amoeba under favorable condition will undergo binary fission. Under unfavorable condition, what will happen? That means when there is lack of food or any adverse condition, they will form a cyst, and they will not form the pseudopodia, and they will form a cyst, and inside there is a protecting cover. You can see the second figure over here. It is a cyst. Inside that itself you can see all the nuclear divides as well as the cytoplasmic division takes place, and many amoebulae are formed. In the third picture you can see there is division, and you can see a lot of amoebulae are inside itself, and only when there is favorable conditions they will burst open and small daughter amoeba or amoebulae comes outside. You can see the fourth picture how the amoeba is the cyst is opening and all the amoeba are coming outside. So this is the binary fission which takes place when in the unfavorable conditions. Okay, so we learnt about binary fission in amoeba, paramecium and euglena. That is simple binary fission. And next we learnt about multiple fission in when there is adverse condition or lack of food. This amoeba undergoes multiple fission. Now coming to the third one, the last one. Today we'll see only these three: asexual reproduction. The third one we are going to learn is budding. this also is a small activity we can do. We can bring the active dry yeast powder from the market. Okay, bring the active yeast powder from the market. Take 50 ml of lukewarm water in a conical flask and then add 5 grams of active dry yeast powder and 10 grams of sugar. So take water, how much? 50 ml of lukewarm water and add 5 grams of yeast and 10 grams of sugar and mix it very well. Okay, and keep that mixture for some time. After that, you keep it in a warm place, and after some time just take a drop and keep it in the clean glass slide. When you see cover, after some time you can see, when you keep it, and after that you take a drop of that water and keep it on the compound microscope. So what you are doing, you are bringing active dry yeast from the market and take 50 ml of lukewarm water, adding 5 grams of yeast and 10 grams of sugar and mixing it and keeping it in the warm place. After that, take one drop of it and keep it under the microscope. You can see the yeast cell performing budding. Okay, you can see them alive. Small bud comes out of many parent cells. That means asexual reproduction occurs by budding in yeast. Okay, yeast cell produce 2 daughter nuclei by mitotic division so as to produce by budding. So you can see what happens. The parent cell, there is a bud formed above of it. You can see here. So what happens, when you take this drop of water and put it under microscope and observe, you can see the parent cell undergoing budding. That is, small daughter buds are formed. That means after that, after the buds is fully grown, they will detach from the parent. You can see this is the yeast cell is called as a parent cell. A small bulge, how this process takes place, a small bulge appears on the surface of the parent cell, and this bulge is actually called as a bud. So one of the two daughter nuclei enters the bud. So the nuclei also divides and one small daughter nuclei enters the bud. So every bud will have nucleus also. So from the parent it will divide and small bud is formed, and that from that parent one small part of the nucleus, that is nuclei, will divide and one part will go into the bud. So even the bud will have the nucleus. Okay, and after sufficient growth, the bud separates from the parent cell and starts to live independently as a daughter yeast cell. So they will make buds. So when the buds are fully grown, okay, after it is attached to the parent, and after they will start to live independently. When they are fully grown, they will separate from their parent and they will grow independent. You can see the diagram of it. See, this is the parent yeast cell, and what's the small bulge comes out, and after that slowly the nucleus, third picture you can see clearly, the nucleus divides and one part of the nucleus has gone to the new bud, and slowly there is a partition between the parent and the bud, and then the bud detaches slowly. They will be joined to one another, and when they are fully grown they will detach from the parent cell. So this is the budding. Okay, so this takes place in yeast cell. A parent is there. So when you take yeast and put it in lukewarm water and take 50 ml of lukewarm water, add 5 grams of active yeast and 10 grams of sugar and mix it and keep it for some time and take one drop of water and observe under microscope. You can see the budding process. What is this? In the parent yeast, a small bud is formed, and the nucleus also divides. The one part of the nucleus enters into the cell, new bud cell, and after favorable conditions, when they are fully grown, they will detach from the parent. So you can see the daughter is, the small buds are formed, they will detach from that. So it is very quick action. So this is the multiplication. This is called as the budding. So children, today we learned what all we learned. We learned about asexual 3 unicellular organisms. We learned about 3 processes. One is binary fission that we saw in amoeba, paramecium and euglena. In amoeba, it takes place in any plane. Okay, in binary, that is division of the parent cell. That's all. In paramecium, we saw it is in the transverse, and in euglena we saw it is in the longitudinal plane. Under unfavorable condition, the amoeba or any other protist will undergo multiple fission. What is this multiple fission? Amoeba will stop producing pseudopodia, and they will form a cyst covering. Inside itself there will be multiplication, nucleus will divide, and cytoplasmic plasm is also dividing, and many amoebulae will be formed inside. And when there is favorable condition, they will burst open and many amoebulae are formed. This is called as multiple fission. And third one we saw was budding. What is budding? I told you, when you take active yeast and put it in lukewarm water and a little bit of sugar and keep it in warm place, budding of yeast takes place. That means from the parent cell a small bulge comes out, that this is called as the bud, and the nucleus of it, nuclei divides, and that will enter into the bud, and every bud will have nucleus also, and when they grow completely, they will detach from the parents. So these three methods we saw of unicellular organisms, that is budding, and we saw about multiple fission and binary fission. All these we learn. Open your textbook if you have a textbook or PDF. Go through all this. These are simple as well as interesting. You can go through this and learn and keep till I start with the next topic. Okay, children. Hope you all have understood. Go through. Okay, take care. Sai Ram.